In our first case example, we will turn black slurry into a low-calorie sweetener. To obtain sugars for fermentation, for instance in a bioethanol plant, traditional pretreatment method for lignocellulose is total hydrolysis, using for instance steam explosion. However, there are alternative methods such as the so-called Organsolv method, in which the three fractions of lignocellulose, the cellulose, hemicellulose, and lignin are separated. Lignin here is obtained in a pure form which is suitable for many applications. Cellulose can be retained in the fiber form, and the hemicellulose comes in the third fraction as mainly monomeric sugars. At best, it can be nearly 30% of the weight of the material.
And here is an example of a composition of the hemicellulose slurry from an Organsolv process developed by a French company, CIMV, from wheat straw. You can see that it contains a significant portion of the C5 sugar, xylose. Also, it contains what remains from the two other fractions, from lignin and cellulose. It also contains all the impurities and some components which originate from lignin, like furfural and phenolics. Also, the organic acids, formic acid and acetic acid, which are used in the fractionation, remain in the C5 side stream, even though part of them can be recycled in the process. So one could, of course, try to ferment this fraction to ethanol, for instance, but it would be maybe more feasible to show that you could directly convert the xylose to more xylose-specific products and maybe with higher yields than you would get ethanol. And we have studied two options here. So you could reduce xylose to xylitol, or you could oxidize xylose to xylonate, xylonic acid. These are single-step reactions. Xylose to xylitol is converted with the enzyme xylose reductase.
Xylitol is an interesting product with a growing market. It's a low-calorie sweetener, and it can be used in chewing gums, candies, food, and pharmaceuticals. But it can even be used as a bio-based component in polymers. The current chemical production method requires high temperatures and toxic catalysts. So Merja, you've studied the difference between two yeast strains. Which were these, and what kind of results did you obtain?
There are differences between yeast strains in the efficiency, how they do this conversion. Baker's yeast Saccharomyces is the most commonly used organism in biotechnical production, but we have also used quite a lot the yeast species Pichia kutriatsevi because it's very robust and it's also acid tolerant, which is important in this case. Here you can see experiments which we have been carrying out now on pure xylose to optimize the strain, and the conversion of xylose to xylitol. If you put to the Saccharomyces host strain 1 copy of the gene, you get xylitol production. 2 copies give you more production, and if you have a multi-copy vector, you still can improve the productivity. But already from this experiment you can see that the Pichia strain is actually better, because 1 copy of the enzyme gene in Pichia gives high productivity, and if you then further increase the copies to 3, you can have quite significant production. In this case, 160 grams per liter could be increased, still providing more xylose. The rates are feasible for industrial production, over 2 grams per liter per hour, and the yields are very high. So it's over 0.9 grams of the product xylitol obtained from 1 gram of xylose. Then we went further, of course, to study how the cells would behave when we are using the Organsolv C5 fraction slurry as a feedstock in the process. And depending how much you feed to the cells this slurry, you can have higher and higher production. And here the best strain titers are over 100 grams per liter. The rates are lower now than we had on pure xylose, but the yields are almost the same as on pure xylose.
Very promising results through optimizing the process. But how were you able to overcome the toxicity of the slurry to the yeast?
What we did was actually to have an enzyme, formate dehydrogenase, which could convert formic acid to CO2. And this gene was introduced to the strain which produced xylitol. Here you can see experiments with 3 different strains. One contains the XR gene only, one only the formate dehydrogenase, and one strain had both of the genes. We used pure xylose and added different amounts of formic acid to the medium. And you can see that xylitol production is only obtained with the strain which has both of the genes, but otherwise the conditions are toxic for the strain, and the conversion of xylose to xylitol does not really happen. The real Organsolv C5 fraction slurry contained 40 grams of formic acid originally. During the process, there is barely no formic acid in the medium. So this additional genetic engineering causes the strain to utilize the formic acid, which then makes the whole process less toxic for the yeast cell and also is crucial for the cell's productivity and success of the process.
This example shows how you can obtain industrially relevant productivity through the choice of strain and the optimization of the pathway enzymes. Also, it shows that challenging materials can be utilized using biotechnology, as we could see how the toxicity of the raw material can be cleaned with the genetically engineered microbes.